We are happy to present through AJOG our work on the pro-inflammatory changes in the maternal circulation, maternal fetal interface, and placental transcriptome in preterm birth. Preterm birth remains a leading obstetric complication due to an incomplete understanding of the multifaceted etiology. This results in important maternal and neonatal morbidities and mortalities. Pregnancy is a transient condition susceptible to stress, hormone imbalances, vascular disorders, and infection, among others, all of which can disrupt the tightly regulated immune pathways in maternal circulation and at the maternal fetal interface that eventually lead to labor. It is known that inflammation is a key component of both physiological and premature induction of labor, where the balance of immune mediators is crucial for timely parturition. Premature immune activation, denoted by immune alterations towards a pro-inflammatory profile, is observed in women with preterm birth. Even knowing this, therapeutic interventions are still lacking, in part due to a scarcity of evidence in the contribution of maternal and placental interrelated compartments. Our primary goal was to obtain an integrated view of the maternal and placental contribution to preterm birth to understand the compartment-specific contribution to the etiology of the syndrome. Briefly, to do this, we looked at gene expression of the placenta, the inflammatory profile at the maternal fetal interface, histological analyses of the maternal fetal interface, maternal circulating immune cell profiles, and inflammatory profiles in the maternal circulation. More specifically, we prospectively recruited 79 women with term or preterm deliveries and collected tissues for RNA sequencing to assess levels of inflammatory mediators and for histological analyses. Blood samples were additionally collected to evaluate the circulating levels of inflammatory mediators and to determine the circulating immune profiles by flow cytometry. Patients from our cohort were largely equivalent, with changes only in maternal age and in relation primiparity. By design, we confirm differences in gestational age at delivery and birth weight. Placental transcriptomic analyses revealed 102 differentially expressed genes upregulated in preterm birth, including known and novel targets. These showed overrepresentation and enrichment of immune-related and inflammatory pathways in preterm birth. We confirmed this through biological gene ontology analyses, gene set enrichment analyses, and pathway enrichment analyses. Circulating maternal immune cells revealed distinct profiles in preterm versus term births, including an increased percentage of CD3 negative cells and monocyte subsets, and a decreased CD3 positive cells, along with Th17 subsets of CD4 positive lymphocytes. Supporting these present findings, we found a decrease in progesterone and an increase in pro-inflammatory C-reactive protein in the plasma, increases in pro-inflammatory mediators such as IL-6 and TNF-alpha in the placenta, with similar findings in the fetal membranes, primarily the amnion, of women with preterm birth, such as monocyte chemoattractant protein 1, C-reactive protein, S100A8, and IL-6, for an overall net pro-inflammatory imbalance in preterm birth. These findings were not distinct between spontaneous and iatrogenic preterm birth, except at the maternal fetal interface, notably in the chorion and at a molecular level, where spontaneous preterm births presented with an elevated inflammatory profile as compared to iatrogenic preterm births, strengthening distinct mechanisms being present at the level of the immune system between these subtypes. Supporting these findings at the maternal fetal interface, we identified an increase in placental abnormalities in preterm birth. In fact, both structural and inflammatory lesions were elevated in preterm placenta. The most frequent inflammatory lesions included maternal inflammatory response and deciduitis, and the most frequent structural defects included excess syncytial knots and excess fibrin in avascular villi. Concordantly, we identified that placentas with inflammatory lesions compared to lesion-free term placentas showed upregulation of inflammatory pathways. Overall, our study brings the field one step closer to new therapeutic targets, with future work looking at components of identified dysregulated signaling pathways and genes. This work can also be used to support currently used therapeutics, such as the use of progesterone, and by contributing to the identification of women with or without inflammation to adapt therapeutic strategies currently in use. Lastly, we've generated a large transcriptional repertoire of changes occurring at the maternal fetal interface in both term and preterm labor. Strengths of our work include equivalent demographics for our population groups, combinations of many techniques as well as several compartments, and fetal sex adjustments in bioinformatic analyses. Whereas some limitations include some patients being in labor at the time of blood collection, preterm birth having an elevated proportion of small for gestational age neonates, and term pregnancies being used as controls. To reiterate, our work sheds light on changes within the immune system in preterm birth on multiple levels and compartments to help identify pregnancies at high risk of preterm birth and to discover novel therapeutic targets. It also identifies novel pathways, enriched and spontaneous versus iatrogenic preterm birth, and identifies transcriptomic changes concerning placental lesions. Though further study is needed, as we continue to integrate findings from multiple interrelated compartments to elucidate their involvement in preterm labor, this represents an important step towards promoting maternal and neonatal health outcomes.